In this webinar, we will walk you through the key aspects of high-throughput screenings in drug discovery and development, as well as our innovative solutions designed to address these challenges. We will start by introducing our company and providing a brief overview of our expertise. And then we will dive into the key factors involved in high-throughput screenings, including some common challenges you might encounter. Next, Melanie will then introduce the technical aspects of our cost-effective solutions for high-throughput RNA sequencing called AccuraCode and AccuraCode TCR. And I will then highlight the versatility of these technologies in research and clinical settings, demonstrating how they actually can enhance efficiency and accuracy. Elena already mentioned it. If you have any questions during the webinar, please feel free to enter them via the Q&A function in Zoom. We will collect all your questions and then answer them at the end in the Q&A session. So let's get started with a few words about the company. Singleron Biotechnology is an international company headquartered in Cologne. And besides having our product development and engineering team there, also the service laboratories can be found here. We are fully committed to single-cell sequencing technologies and have developed a very comprehensive portfolio of different single-cell RNA-seq methods. And they are available both as kits as well as services. We offer solutions for basically every step of the workflow, starting from preservation and also dissociation of fresh tissue, to our proprietary chip-based cell partitioning solution, and finally also to bioinformatics tools for a customized data analysis. However, this presentation actually will not so much focus on single-cell sequencing, but we will rather discuss how we adapted the core technology of our single-cell sequencing method, which is based on a microwell chip for capturing individual cells and barcoded beads for the mRNA capture to enable its application in bulk mRNA sequencing, and particularly for high-throughput experiments such as large-scale compound screenings, for example. Before Melanie dives into the technical details of our products, I would like to set the stage by providing an overview of the key steps in drug discovery and development in the process of drug discovery and development, along also with some major challenges that we might face. So as you might know, the journey of drug discovery and development is very complex and multifaceted, aimed at identifying and also bringing new therapeutics to the market. It involves several critical stages, and it starts with the target discovery. In this first step, the goal is to identify a biological target that is not only relevant to the disease, but it also needs to be druggable at the same time. Following that, we move into hit identification and lead optimization, where high-throughput screening and also computational methods are employed to find compounds that interact with the target and also produce a desired effect. Promising candidates then further proceed to in vitro and in vivo preclinical studies to assess their efficacy, the toxicity, and also the pharmacokinetics. If the drug really successfully passes all these studies, it then enters the phase of clinical trials to evaluate safety, efficacy, and also the dosage. And this is actually the most time-consuming and also the most expensive phase of the whole process. So once these clinical trials are successfully completed, regulatory agencies will review the data before then hopefully granting also approval for market release. Despite several advancements in the technology and also in data-driven approaches, the drug discovery and development journey still remains highly expensive and also very time-intensive. So it typically takes around 10 to 15 years with a high attrition rate. And overcoming these challenges really requires innovative screening methods, but also biomarker-driven strategies and advanced analytical tools to further improve the efficiency and also the success rates. One of the very first and also most crucial steps on the path to drug discovery, as I've already mentioned before, is high-throughput screening. While it actually might sound quite straightforward, it is actually a really highly time-consuming and also labor-intensive process. And it also has significant consequences for all the other steps that follow. At its core, high-throughput screening is designed to identify bioactive compound from chemical libraries. But it's just not as simple as just picking out the first promising compound you find. It is really a highly strategic process where you gradually narrow down the list of potential candidates with each stage. So in the initial screening phase, we are talking about testing thousands to millions of compounds. And the goal is to really uncover compounds that show any sign of activity against the target molecule. Of course, this phase comes not without its challenges. It has a really high false positive rate. So meaning that while many compounds might appear promising at first, only a small fraction will actually make it to the next round. These initially identified hits are then retested, mostly under the same conditions, to weed out the false positives. Also, this is a very critical step that significantly reduces the number of candidates that move to the next round, which will be the concentration response screening. Here we test the before confirmed hits at various concentrations to assess their efficacy. And this phase is really all about identifying which compounds show real and also reliable potential. And these are the ones that then remain as promising lead candidates. By the end of the whole process, usually less than 1% of the original chemical library remains. So it's really a journey of elimination, refinement, and also discovery that sets then the stage for the next critical steps in drug discovery and development. Since high-throughput screening plays such a pivotal role in shaping the entire drug discovery and development process, a careful planning of the experimental design is actually absolutely crucial. So one important aspect is, for example, the selection of the appropriate starting material. So here, cell lines are, of course, a popular choice due to their reproducibility and also the ease of use. But there are actually other options that provide a closer representation of real-world conditions. So spheroids and organoids, for instance, they are far more complex, but they also offer more physiologically relevant models. So once we've selected our starting material, the next key step is determining the readout for the screening process. And the readout is actually the lens through which we will view the data and then also interpret our findings. So typically, high-throughput screening assays just focus on only a single parameter, which makes the data easier to analyze and also ensures that we get reliable and robust results. It could be anything from measuring cell viability, for example, to monitoring protein expression using cutting-edge imaging techniques, or also assessing a specific cellular function like a substrate depletion, for example. This really depends on the experiment's goal and also on the design of the experiment. The accuracy and also the sensitivity of this readout is, of course, crucial. So it's really what enables us to identify meaningful hits that can lead in the end to groundbreaking discoveries. So what if we could actually move beyond the traditional single parameter readout and capture a broader and also a more comprehensive picture while still maintaining the same level of accuracy and sensitivity? So at first glance, using transcriptional readout for high throughput screening might seem highly costly and also less targeted, but in reality, it actually brings with it quite some incredible advantages. So in the next minutes, Melanie will first give you some insights in how our AccuraCode bulk RNA sequencing product portfolio works and why it is especially powerful in high throughput environments. And then I will dive into some concrete examples showing you how incorporating transcriptional readouts doesn't only accelerate your high throughput screening assay, but it also can enhance other critical stages of the drug discovery and development process. So with this, I will now hand over to Melanie, who has all the technical information for you.
The AccuraCode portfolio is designed for high throughput transcriptomic analysis, and it's a plate-based assay that combines barcoding technology to with bulk RNA-seq to generate libraries from many samples at the same time in one tube. So it allows simultaneous library preparation of up to 384 samples in just a single tube. This means that we're actually reaching an increased throughput with reduced processing cost and also time. So that's, of course, very exciting for everybody working in the lab. And it is a streamlined workflow that allows for either manual or automated operation, so it's also flexible in that regard. And we offer the whole transcriptome profiling, AccuraCode RNA, and also the TCR profiling solution, which is called AccuraCode TCR. So I'm first going to now tell you a little bit about the principle of the technology. So the essential step of the technology is we have beads that are covered with oligos that end in poly-Ts, which allow us to capture polyadenylated RNA, mRNA. And then after that, during the reverse transcription step, we can actually barcode the samples. And then afterwards, because they are then barcoded, each sample separately, we can then pool them, do the cDNA amplification, and construct our library and everything in one sample, in one tube, not in one sample. And here on the right, you can see the AccuraCode RNA, like in a diagram of the workflow. So for the AccuraCode RNA kit, we start with paramagnetic beads, which are covered in oligos that also contain the barcode, the sample barcode, and the UMI for the quantification of the RNA, of the transcripts. And then it ends in a poly-A tail, which allows the capture. During the reverse transcription step, then we will, through the poly-A, we will first capture the mRNA as indicated here. And then in the reverse transcription step, we on one hand barcode it, and at the same time transcribe the RNA into cDNA. And then the cDNA we can then pool all together for the different samples into one tube and perform the library preparation. The AccuraCode TCR is essentially a variation of that workflow. It is fairly similar, and I'm not going to go through all the details in which they differ. The main difference is that for the AccuraCode TCR, that the barcoding bead, the bead actually is not a barcoding bead. and the barcode instead will be introduced during the reverse transcription step on the TSO primer. And then after that, the reverse transcription also results in a barcoded in barcoded cDNA, which we can then pool and amplify and then prep in the libraries. Okay, so here you can see an overview of the AccuraCode workflow. It essentially allows you to go directly from cells to libraries without having ever to isolate RNA if you want to. So our sample input materials are, as I just said, cells. You can directly lyse the cells on the 96 well plates and then capture the mRNA in the same plate afterwards. So for this, you can either use adherent cells or suspension cells, but you can also, of course, provide RNA. That's not a problem at all. But since we are pooling the samples later on, it is important that the sample input is normalized. So we need to start with the same amount of cells or the same amount of RNA for each sample so that they are at the end equally represented. Okay, after this, from then we barcode the RNA during the reverse transcription step. Then we can pool all our samples together, amplify the cDNA, and then amplify the library. Of course, here the workflows will differ, since for the transcriptomic part, we are just like normally constructing an RNA library, while for the TCR AccuraCode, we will need to first enrich for the TCR sequences within our mRNA sample. And then after enrichment, we can create the libraries. And then during the sequencing step, we will our libraries are they are handed in for paired end sequencing, and the first read is used to resolve the barcode, so to know from which sample the information comes, and then the second read will actually provide the information about the transcript sequences that we captured. Okay, so now I wanted to show you a little bit of performance data for AccuraCode TCR. So what you can see here is so we wanted to see how many cells, how many T cells in a mixed sample, for example, we need to be able to detect them with AccuraCode TCR. And so here you can see what we did is we used PBMCs and CCRF, that's a cell line, and the cell line is known to contain only a single clonotype. And by mixing them, and so we mixed them at different fractions. So we mixed 10,000 PBMCs and added only a single cell from the cell line, up to half equal amounts of both. So these mixed samples we used then through the workflow. So we lysed them, we performed RT amplification, quality control, sequencing, and analysis. And then we wanted to know, so do we see the expected results? So here, since we know that CCRF only, like we can detect it in the mixture based on its like a single clonotype. So what we can, here on the y-axis, you can actually see the CCRF proportion in the mix. And you can actually see that, as expected, there's like in between, when we have 2,000 CCRF cells compared to 1,000 CCRF cells present in the sample, we do see roughly a twofold decrease, and then the same when we go from 1,000 CCRF cells to 500, and then we do see about fivefold drop in CCRF representation in the sample when we only have at the end 1,000 CCRF cells. So this means that AccuraCode allows us to detect rare T cells that are even present as low as 1% of the total sample. Okay, and then I wanted to share with you some results on a research collaboration that we're working on together with the uni clinics in Cologne and Münster. So this project is trying to detect circulating T cells in a liver disease that I unfortunately can't disclose because it is still ongoing research. So we used AccuraCode TCR to analyze the T cell receptor repertoire. And in this case, we worked with Buffy code samples of 96 different patients, and it's actually a progressive liver disease, so we can actually look at different stages. So here now we're comparing the control versus stage 1 and stage 2. So the healthy control versus stage 1 and stage 2. Here the larger, so essentially you can take two things out of this graph. So on one hand, the more different bars are present means the more different T cell clonotypes were detected. So we can see that in the control, there's a lot less than in the stage 1 and in the stage 2 group. And on top of that, we can also, on the y-axis, we can also see the number of clonotypes. So the higher the bar, the more clones with the same T cell clonotype were detected. So what we can see here is that there's a significant increase of number of T cell clonotypes and clone counts with disease progression. So something changes in the TCR repertoire during the disease. And then we also looked at the analysis of the CD3 gene recombinations. So these are graphs of the healthy controls and then of the, I believe it was the stage 2 group. And essentially what this type of analysis allows you to see is which type of gene combinations were detected in different samples, but repeatedly. Because the T cell receptor is made up of different genes that are recombined. And so here they were actually able to identify a T cell receptor beta chain that was specific to the stage 2 group. And so this is very exciting and ongoing research. And yeah, from here, I'm gonna just talk a little bit more about applications areas. So I think, as you've seen, AccuraCode is a high-throughput transcriptomic screen platform. It allows, for example, application in drug screening, biomarker discovery, and drug development, as well as gene and cell therapy approaches, especially using the TCR solution. And from here, I'm going to hand the mic back to Jacqueline so she can finish it up.
Okay, thanks, Melanie. So now that we've covered all the technical details, and also Melanie also introduced one very exciting example, I would also like to focus on some examples of how these tools can actually significantly improve the outcome of your drug development process. So let me walk you through an experiment that we conducted internally. In a small but yet insightful study, we treated A549 cells with increasing concentrations of 3 different compounds, ranging from 1 micromolar to 50 micromolar, with DMSO as the negative control. The RNA-seq cells, which are here visualized as a heat map, provide us with a deep view into the cellular responses to these compounds. So the heat map clearly shows how compounds P and T exhibit almost identical transcriptional patterns to DMSO, which indicates that they do not induce significant changes. And in contrast to that, compound B displays a completely distinct and unique transcriptional profile. So these differences can really offer valuable insights into the efficacy of these compounds in your research. And with subsequent analysis, we can additionally also dive deeper into differentially regulated pathways, for example, which allows us to gain a more comprehensive and also precise understanding of complex biological processes. And that's actually not all. These analyses also provide us with a very detailed, time-resolved and concentration-dependent insight into the expression of patterns of individual genes or also entire gene families that might be of particular interest. So these various analysis options provide you with much more than just the identification of an active compound during high-throughput screening, which actually also brings me directly to another advantage of having transcriptional data as a screening readout. So let's reconsider how we approach the understanding of diseases in the context of drug discovery. Traditional models often treat disease as a simple on-off switch, but actually in reality, we all know that diseases are much more complex. So they evolve over time. They can present differently in individual patients, and they have diverse manifestations. So that really all challenges our understanding. And this is actually where transcriptional readouts and particularly RNA-seq methodologies as AccuraCode that we just presented step in. So you're not just limited to this one-dimensional perspective. You can really track the progression of the disease across different stages. You can uncover molecular signatures that provide really a deeper understanding of complex biological effects. So this level of detail allows you to really tailor your drug discovery process in a way that it accounts for the disease's complexity and diversity. A transcriptional readout in high-throughput screenings also offers a powerful way to uncover the mode of action of compounds. And this also already early in the process. So by analyzing changes in gene expression, we can gain insights into the molecular mechanisms that a compound is affecting within a cell. And unlike traditional assays that may only measure a single cellular response, the transcriptional profiling allows to capture a broader spectrum of cellular activity and really revealing how a compound influences various biological pathways. This allows us to identify whether a compound is acting through known pathways. We can also uncover off-target effects, or we can even detect novel mechanisms of action. And by catching these effects early on, we can make a more informed decision on which compounds to pursue. And of course, this saves us a lot of time and resources throughout the whole process. An example of how researchers use the combination of AccuraCode and AccuraCode TCR to elucidate the mode of action of compounds is shown here. In this study, a T-cell containing organoid model from bladder cancer patients was developed to evaluate the immunosynergistic effects of platinum drugs in combination with immune checkpoint inhibitors. So the goal was to identify immune-related mechanisms involved in this combination therapy. And to explore T-cell related mechanisms, the researchers first performed bulk TCR-seq and then also added RNA-seq on these bladder cancer organoids treated with the platinum drug PD-19 and PD-1 inhibitors. The results revealed that PD-19 treatment significantly amplified TCR clones in the organoids compared to the control and anti-PD-1 group alone. And moreover, the combination of PD-19 and immune checkpoint inhibitors also further enhanced the enrichment of TCR clones. And with the help of the transcriptome analysis, the researchers could additionally find that the combination is not only promoted T-cell activation, but it actually also improved antigen presentation. So this study really is a nice example of how transcriptional readouts like RNA-seq and also TCR-seq can help to uncover the mode of action of compounds, revealing not just their immediate effects, but also their influence on very complex systems like immune mechanisms. There are actually several more examples on how AccuraCode and AccuraCode TCR as transcriptional readouts can help you to accelerate your drug discovery journey. So the tools can help you to minimize false positives rates, which really ensures you that you are on the right track from the very beginning. By using this approach, you can also detect, as already mentioned, off-target effects and toxic responses early on, so long before they become major roadblocks. And in my eyes, also the flexibility of the readout is another key advantage. So it can be easily adapted to suit different experimental setups, making it a very versatile tool for your specific needs. And by obtaining robust and reliable data in the early stages, you also lay, of course, the foundation for optimizing preclinical and clinical phases down the line. So with our cutting-edge AccuraCode high-throughput RNA sequencing portfolio, we enable multiplexed RNA library preparation. By eliminating the need for time-consuming RNA extraction and streamlining also the library prep into a single tube for multiple samples, you really can save significant time and resources, resulting then finally in consistency, reproducibility, and also considerable cost savings, of course. So I really hope that we've been able to show you how AccuraCode can offer value at multiple steps, making your journey through drug discovery and development not only faster, but also more reliable and cost-effective.